we can program it to cut wherever we want. That sentence appears in a 2012 interview with Jennifer Doudna, recorded months before the paper that would reshape molecular biology. She was describing a bacterial immune system, a mechanism that bacteria have used for over 1 billion years to recognize and destroy viral DNA, and explaining, with the calm precision of someone who has not yet grasped the scale of what she is holding, that this mechanism could be turned into a tool, a programmable tool, a tool that edits genes. Any gene, in any organism, with a precision that no previous technology had achieved. 6 years of work by 2 women on 2 continents, distilled into 9 words that made the rest of biotechnology obsolete overnight. The paper was published in Science in June 2012. Within a year, the tool had been used in human cells, mouse embryos, zebrafish, and rice. Within 5 years, it had spawned an industry worth billions. Within 8 years, it had earned a Nobel Prize. The patent, however, went to someone else. The system is called CRISPR-Cas9. The acronym stands for Clustered Regularly Interspaced Short Palindromic Repeats, a name that accurately describes the structure but conveys nothing about the function. The sequences were first noticed in 1987 by a Japanese researcher named Yoshizumi Ishino, who found them in E. coli while studying a completely unrelated gene. He noted the repeating pattern. He did not know what it did. Nobody did, for nearly 2 decades. In 2005, a bioinformatician in Spain named Francisco Mojica published the insight that the sequences between the repeats matched known viral DNA. The bacteria were keeping records. They were storing fragments of every virus that had ever attacked them, filed between repetitive spacer sequences like entries in a criminal database. The function is elegant. When a virus appears again, the bacterium transcribes the stored fragment into a short piece of RNA, which guides a protein called Cas9 to the matching sequence in the invading DNA. Cas9 cuts both strands. The virus is destroyed. The system is ancient, over 1 billion years old, and until 2012 of interest primarily to microbiologists studying bacterial immune defenses. Mojica had tried to publish his findings multiple times before 2005. Nature rejected the paper. So did the proceedings of the National Academy of Sciences. The mechanism was too obscure. The bacteria were not interesting enough. When Doudna and Charpentier transformed CRISPR into a tool, Mojica's name did not appear on the paper. His contribution, identifying the biological function, received no patent, no company, and no prize. What Doudna and her collaborator Emmanuel Charpentier demonstrated in their science paper was that this system could be reprogrammed. The collaboration had begun in 2011, when the two met at a conference in Puerto Rico. Doudna was a structural biologist at UC Berkeley, methodical, precise, deeply embedded in the RNA world. Charpentier was a microbiologist working in Sweden, studying the CRISPR system in Streptococcus pyogenes, the bacterium that causes strep throat. Charpentier had identified a critical component, a short RNA molecule called tracer RNA that was essential for Cas9 to function. Together, they fused the 2 RNA components into a single guide molecule and demonstrated that by designing a synthetic guide RNA matching any target DNA sequence, they could direct Cas9 to cut at any location in any genome. The implications were immediate and enormous. Gene editing had existed before. Zinc finger nucleases and talons could cut DNA at specific sites, but these earlier tools required weeks of protein engineering for each new target. They were expensive, slow, and required expertise that most laboratories did not have. CRISPR was different. A graduate student could design a guide RNA in an afternoon, order it from a supplier for a few $100s, and have results within a week. The technology democratized genetic engineering. It also created a problem that no amount of scientific elegance could solve. Who owned it? 6 months after the Doudna-Charpentier paper, a researcher at the Broad Institute in Cambridge, Massachusetts, filed a patent application. His name was Feng Zhang. He was 30 years old, a prodigy who had been recruited to the Broad and MIT with his own laboratory while still completing his postdoctoral work. Zhang had been working on CRISPR independently, using a different Cas9 variant and a different delivery system. His contribution was specific and significant. He had demonstrated that CRISPR-Cas9 worked in eukaryotic cells, the cells of animals, plants, and humans. Doudna and Charpentier had shown the system worked in a test tube, using purified components in a cell-free environment. Zhang had shown it worked inside living human and mouse cells, editing genomes in their native context. The distinction sounds technical. In the language of patent law, it was the difference between describing a principle and reducing it to practice. It was worth billions. Zhang's patent application was filed with a request for accelerated review, a procedure available at the U.S. Patent Office for an additional fee. The Broad Institute paid the fee. The strategy was deliberate. The Institute's Patent Council later acknowledged that the accelerated track was chosen specifically to establish priority before Berkeley's pending application could be examined. The application moved through the system in months rather than years. In April 2014, Zhang was granted a patent covering the use of CRISPR-Cas9 in eukaryotic cells. Berkeley's application was still under review, caught in the normal pace of patent examination, which can take 3 to 5 years. The timeline created a paradox that the patent system was not designed to resolve. The person who published the foundational discovery did not hold the patent for its most valuable application. The person who filed second, with an accelerated request and institutional backing, held the rights to the technology's commercial future. The commercial stakes were not abstract. Before the patent was even granted, both sides had founded companies. Doudna co-founded Caribou Biosciences in 2011, which later spun off Intellia Therapeutics, focused on CRISPR therapeutics. Zhang co-founded Editas Medicine in 2013, backed by the Broad Institute's patent position. Charpentier co-founded CRISPR Therapeutics, headquartered in Switzerland. Venture capital poured in, hundreds of millions of dollars in the first round alone, distributed across companies whose entire valuations depended on which patent claims survived legal challenge. By 2015, the three companies had a combined market capitalization approaching $3 billion. None of them had a product. None had completed a clinical trial. The value was not in results, it was in rights. The science had fractured into competing corporate entities before it had finished being published in journals. Researchers who had once collaborated freely now communicated through attorneys. In January 2016, the U.S. Patent Trial and Appeal Board declared a patent interference proceeding, a formal inquiry into whether the Berkeley and Broad claims overlapped. The question was narrow but consequential. Was the use of CRISPR in eukaryotic cells an obvious extension of the Doudna-Charpentier discovery, or was it a separate invention? If obvious, Berkeley's earlier publication date would give them priority. If separate, Zhang's patent could stand alongside it. The legal teams on both sides included some of the most expensive patent attorneys in the country. The stakes, control over licensing fees for every CRISPR-based therapy, agricultural application, and research tool, justified the cost. The proceeding lasted 13 months. Expert witnesses testified about molecular biology, experimental timelines, and the definition of obviousness. Berkeley argued that once the fundamental mechanism was published, any competent molecular biologist could have made it work in human cells, and several did, within weeks of the paper. The Broad argued that the step from purified components in a test tube to functional editing inside a living nucleus involved unpredictable challenges that Zhang had solved through genuine invention, not routine application. In February 2017, the board ruled in favor of the Broad Institute. The decision hinged on reduction to practice, the difference between describing an idea and demonstrating that it works in the context that matters. Doudna and Charpentier had described the tool. Zhang had made it function in the cells that the world wanted to edit. The board found no interference in fact. The two inventions were distinct enough to coexist. Berkeley appealed. In September 2018, the Federal Circuit upheld the ruling. The patent landscape solidified. Doudna published first. Zhang patented first. Both claims survived, covering different territory. The science said one thing, the law said another. The money followed the law. The story has a structural echo that runs through the entire second month of this channel. When a finding is real, but the system that processes it, whether medical, ecological, or legal, is built to reward something other than truth, the finding survives, but its meaning changes. A cure for ulcers existed for a decade before the system acknowledged it. A reef died while the tourism industry sold tickets to see it alive. And now, a technology that could cure genetic disease was carved up by patent attorneys before the first patient was treated. The patent system does not ask who deserves credit. It asks who reduced the invention to practice. The Nobel Committee does not ask who filed first. It asks who made the fundamental discovery. The financial markets do not ask either question. They ask who holds the license. The answers point to different people. None of the systems is lying. They are answering different questions, and the patient with sickle cell disease in Mississippi is not part of any of them. The Nobel Prize in Chemistry was announced in October 2020. It went to Jennifer Doudna and Emmanuel Charpentier for the development of a method for genome editing. Zhang was not included. The omission generated debate in the scientific community that is not settled.
Hold it steady right there.
Supporters of the committee's decision pointed out that Doudna and Charpentier had made the conceptual leap, transforming a bacterial curiosity into a universal tool. The Nobel traditionally rewards fundamental insight, not incremental application. Supporters of Zhang argued that the conceptual leap was incomplete without the demonstration that the tool worked where it mattered, in the cells of living organisms. Making something work in a test tube and making it work inside a human nucleus are separated by problems that could not have been predicted from the original paper. The committee did not explain its reasoning in detail. It rarely does. The citation emphasized the discovery of genetic scissors and their transformative potential. Zhang's name appeared nowhere in the announcement. Doudna, asked about Zhang in interviews, was measured. She acknowledged his contributions while maintaining that the foundational work was hers and Charpentier's. Zhang, asked about the Nobel, said little in public. The Broad Institute released a statement congratulating the laureates. The statement did not mention the patent. The Nobel rewarded the idea. The patent rewarded the application. The prize carried prestige, a gold medal, and roughly $1 million shared between two people. The patent carried licensing revenue from every laboratory, every biotech company, and every clinical trial that used CRISPR-Cas9 in human cells. The economics were not comparable. By 2025, the global CRISPR market was valued at over $10 billion. Clinical trials using CRISPR-based therapies were underway for sickle cell disease, beta thalassemia, certain cancers, hereditary blindness, and high cholesterol. The first CRISPR therapy, Casgevy, developed by CRISPR Therapeutics and Vertex Pharmaceuticals, received regulatory approval in the United Kingdom in November 2023 and in the United States the following month. The therapy treats sickle cell disease by editing a patient's own stem cells. The treatment process takes months. Stem cells are extracted, edited in a laboratory, and reinfused after the patient's bone marrow has been destroyed with chemotherapy. The cost per treatment exceeds $2 million. In the United States, fewer than 100 patients receive the therapy in its first year. Sickle cell disease disproportionately affects people of African descent, a population that, in the American healthcare system, is least likely to have access to a $2 million procedure. Charpentier's company developed the product. Zhang's patent covers the underlying method used in human cells. Doudna's discovery made all of it possible. The revenue flows through legal channels that none of the three scientists fully control, and the patients who need the therapy most are the farthest from affording it. The question that the CRISPR story leaves unanswered is not who deserves the credit. That question has been answered, differently, by three separate systems, each operating by its own rules. The question is whether a discovery of this magnitude should be ownable at all. Bacterial immunity evolved over billions of years. The repeating sequences were first noticed by Ashino in 1987. Mojica identified their function in 2005. Doudna and Charpentier reprogrammed the system in 2012. Zhang made it work in human cells months later. At which point in that chain does a natural phenomenon become private property? The Patent Office has an answer. The Nobel Committee has a different one. The bacteria, presumably, have no opinion. We can program it to cut wherever we want, Doudna was right. The question nobody thought to ask in 2012 was who would program it and who would collect the fee. Next time, a species was declared extinct. 40 years later, it reappeared, and the data that saved it belonged to the people who had been thrown out for collecting it.